In the quiet soil beneath your feet, in the darkness where light never penetrates, an ancient conversation is taking place. It is a dialogue conducted not in words but in molecules, not through sound but through chemical signals, passing along threads so fine they are invisible to the naked eye. This is a conversation that predates human language by hundreds of millions of years, a partnership so fundamental to life on land that without it, the world you know could not exist. Welcome to an exploration of unlikely partnerships in nature, of relationships that transcend the boundaries we imagine separate one organism from another, of collaborations so successful they have persisted and evolved across unimaginable spans of time. As we journey through these partnerships, let yourself relax into the rhythm of these ancient stories. Stories of cooperation and interdependence that have shaped the living world. Take a deep breath, settle into a comfortable position, and allow yourself to drift into the hidden realm beneath the surface, where partnerships older than mountains continue their work without pause, without recognition, sustaining the green world above. Close your eyes, if you wish, and imagine a forest. Perhaps it is a forest you have walked through, where shafts of sunlight pierce the canopy and dapple the forest floor, where the air smells of moss and decomposing leaves, where birds call from unseen perches, and the ground beneath your feet feels soft with accumulated leaf litter. The trees rise around you, Douglas firs and birches, hemlocks and pines, their trunks solid and reassuring, their branches reaching skyward toward the light. What you see, what naturally draws your attention, are these visible parts, the trunks thick as your body or thicker, the branches spreading in fractal patterns, the leaves catching sunlight and transforming it into energy through the quiet miracle of photosynthesis. But this visible forest, magnificent as it is, represents only a portion of what exists here. For every tree you can see, there is an equivalent tree below ground, a mirror image of roots, spreading as widely as the canopy above, searching for water and anchoring the massive structure against wind and gravity. And beyond even these roots, spreading through the soil, in an intricate three-dimensional web that defies easy visualization, lies another forest entirely. A forest of fungal threads, a network connecting tree to tree, plant to plant, creating a living system of exchange and communication that we are only beginning to understand. These fungal threads, called hyphae, are impossibly thin, far finer than the roots they surround. A single fungal hypha might measure only a few micrometers in diameter, perhaps 1/20th the width of a human hair, requiring a microscope to see. The individual threads are delicate, fragile things that would snap if you tried to pull them from the soil. But what fungi lack in girth, they compensate for in extent. In sheer proliferation, the hyphae branch and rebranch, growing through soil particles, around pebbles, between root hairs, through channels in the soil that are invisible to the naked eye, creating a network of astonishing density. In a single teaspoon of healthy forest soil, there may be several miles of fungal hyphae, a length that seems impossible given the small space they occupy. Think about that for a moment. Miles of living thread, branching and rebranching, all packed into a space no larger than the bowl of a spoon. Scale that up to a square meter, to a hectare, to an entire forest, and you begin to grasp the magnitude of the fungal presence. This is not a collection of individual, isolated organisms each pursuing its own survival. This is a web, a network, a living internet threading through the soil, and the trees are connected to it, plugged in, constantly exchanging information and resources through these underground cables. The partnership between trees and fungi is called mycorrhiza, a word that comes from Greek roots, meaning fungus and root, a simple name for an extraordinarily complex relationship. It is a relationship of stunning ubiquity. Somewhere between 80 and 90% of all plant species on Earth form mycorrhizal associations. Think about walking through a meadow, or a forest, or even a city park. Nearly every plant you see, from the towering trees to the smallest wildflowers growing in the shade, maintains this partnership. In forests specifically, the percentage approaches 100%. Every tree, every shrub, nearly every plant you encounter has co-evolved with fungi, depends on fungi, has shaped its entire biology around the expectation that fungi will be present. This is not an occasional alliance, not a relationship that forms only under certain conditions or in certain places. This is foundational to how terrestrial ecosystems function, as basic to plant life as photosynthesis itself. And the partnership is ancient. Fossil evidence from Scotland, from a remarkable geological formation called the Rhynie Chert, shows plant tissues with unmistakable fungal structures inside them. These fossils formed 407 million years ago, preserved in extraordinary detail, when silica-rich water from ancient hot springs flooded a wetland ecosystem and entombed everything living there, crystallizing them in place at the cellular level. When paleobotanists slice these rocks thin enough to examine under microscopes, they can see individual plant cells, the walls and internal structures, preserved as if the plant had been alive yesterday. And within these cells, within the tissues of plants so primitive they had no true roots, only simple underground structures called rhizoids. There are fungal structures, arbuscules, the tiny branching structures that allow nutrient exchange between fungus and plant. These formations are identical to arbuscules in modern mycorrhizal relationships. The partnership we see today, operating in forests and fields around the world, is the same partnership that existed when plants first colonized land. When plants first ventured out of the water, adapting to terrestrial existence, learning to stand upright without the support of water, developing systems to prevent desiccation in the air. Fungi were there, partnering with them, helping them extract nutrients from the rocky surfaces that would eventually become soil. It is entirely possible, even probable, that without these fungal partnerships, plants could not have successfully colonized land at all. The fungi may have been the key that unlocked terrestrial ecosystems, the missing piece that allowed green life to spread across the continents. What makes this partnership work? What do the two partners exchange that creates such a successful, enduring collaboration? The plant, whether tree or shrub or herb, gains something it desperately needs, but finds difficult to obtain on its own: nutrients. Specifically, nutrients that exist in the soil in forms or locations the plant cannot easily access. Roots are remarkable structures, capable of growing through soil, absorbing water, anchoring the plant, sensing gravity and moisture gradients, responding to chemical signals. But roots have fundamental limitations imposed by their size and structure. They can only explore a tiny fraction of the soil around them. A root system, no matter how extensive it appears, actually occupies less than 1% of the soil volume in which it grows. Picture a tree with a root system 30 feet across, extending 10 feet deep. That sounds like a lot of root. But the soil volume within that space is mostly empty space, air pockets between particles and the roots themselves. Even with their myriad branch tips and root hairs, contact only a small fraction of that volume. The soil is a three-dimensional matrix, and roots can only access nutrients that happen to be immediately adjacent to their surface. This is a problem because many nutrients plants need exist in very low concentrations, scattered throughout the soil. Phosphorus, in particular, is often the limiting factor in plant growth, the nutrient most likely to constrain how large or how quickly a plant can grow. It is present in soil, often in substantial total amounts, but it is not mobile. Unlike nitrogen, which can move through soil and water as dissolved nitrate, phosphorus binds tightly to soil particles. It does not dissolve readily in water and diffuse toward roots. It must be encountered, stumbled upon by a root or root hair, and roots, occupying less than 1% of the soil, miss most of it. A plant without mycorrhizal fungi is like a person trying to harvest a field of grain while standing in one spot, able to reach only what grows within arm's length, while the rest of the field remains untouched, unharvested, inaccessible. Fungal hyphae solve this problem with elegant efficiency. Those thread-like structures, far thinner than the finest roots, 10 times thinner, 20 times thinner, can penetrate spaces roots cannot reach. They extend outward from the root surface, radiating through the soil in all directions, creating an enormous surface area for nutrient absorption. A single fungal hypha can extend 10 centimeters or more from a root tip, and given that a single tree might have billions of mycorrhizal tips on its root system, the total extent of fungal exploration becomes truly astronomical. These hyphae effectively increase the root's foraging range by orders of magnitude. Through these threads, the fungus absorbs phosphorus, nitrogen, micronutrients like copper and zinc, water, all the things the plant needs, and transports them back to the root, delivering them directly into plant cells. Experiments using radioactive tracers have shown the pathway clearly, demonstrating beyond doubt that this transfer is real and substantial. A phosphorus atom, absorbed by a hypha 20 centimeters from a root, can appear in the plant tissue within hours, transported along the fungal thread, passed through the interface between fungus and plant, and incorporated into plant molecules. The fungus functions as an extension of the root system, a far more efficient extension than roots could create on their own. Studies have shown that mycorrhizal fungi can provide up to 80% of a plant's phosphorus. 80%. Without the fungus, the plant would have access to only 1/5 of the phosphorus it actually uses. Thanks to this fungal network, a plant can access nutrients from a soil volume 50 to 1,000 times larger than its roots alone could explore. In return, the plant provides something the fungus cannot make for itself. Fungi are heterotrophs. They cannot photosynthesize, cannot pull carbon dioxide from the air, and use sunlight to transform it into organic molecules. They require carbon already fixed, already in the form of sugars and other organic compounds, and plants, through photosynthesis, are carbon-fixing machines. In every leaf, in every needle, Sunlight strikes chlorophyll molecules and initiates a cascade of reactions. Water molecules split. Carbon dioxide from the air combines with the released hydrogen. Oxygen escapes back into the atmosphere, and sugar molecules form: glucose and fructose and sucrose. The fundamental building blocks of life, the currency of biology. A portion of these sugars, as much as 20% of everything a plant photosynthesizes, travels down from the leaves, through the stems, into the roots, and is delivered to the fungal partner. This is not a trivial investment. 20% of a plant's photosynthetic production represents an enormous allocation of resources, equivalent to the plant working 1 day out of every 5 exclusively to feed its fungal partners. But the investment pays dividends. With those sugars, the fungus grows, explores new soil, absorbs more nutrients, and delivers more resources back to the plant. The partnership becomes self-reinforcing, a positive feedback loop. The better nourished the plant, the more sugars it produces. The more sugars the fungus receives, the more it grows and the more nutrients it delivers. It is a biological investment strategy that has proven spectacularly successful, persistent across 400 million years of evolution, surviving mass extinctions and ice ages, adapting to every terrestrial environment from tropical rainforests to Arctic tundra. There are 2 major types of mycorrhizae, distinguished by how the fungus interacts with the root cells, by the architecture of their interface. Ectomycorrhizae, common in temperate forests, form a sheath around the root tip, like a glove surrounding your hand. The fungal threads weave together into a dense layer that completely envelops the root tip. And from this sheath, hyphae extend both outward into the soil and inward between the root cells. These internal hyphae do not penetrate into the cells themselves. Instead, they grow in the spaces between cells, forming a structure called the Hartig net, named after a German forester who first described it. The Hartig net is where the exchange happens, where the fungus delivers nutrients into the spaces between cells and the plant cells absorb them, where the plant secretes sugars that the fungus absorbs. The root itself transforms in the presence of the fungus, becoming more branched, thicker, often changing color from white to brown or black, taking on a coral-like appearance that distinguishes mycorrhizal roots from non-mycorrhizal ones. You can see ectomycorrhizae if you dig carefully around the base of a pine or oak tree. The root tips will be stubby, branched, colored, dark with fungal tissue, utterly different from the pale, pointed tips of roots, without fungal partners. Arbuscular mycorrhizae, the more ancient form evolutionarily, found in about 70% of plant species, take a more invasive approach. Here the fungal hypha penetrates into the root cells themselves, though it does so without killing the cell or even breaking its membrane. The hypha pushes against the cell membrane, invaginating it, like pushing your finger into a balloon without popping it. And once inside the cell, the hypha branches into intricate tree-like structures called arbuscules. These arbuscules, named for their resemblance to little trees, are the defining feature of this type of mycorrhiza. They last only a few days before the plant digests them, and the fungus grows new ones, a constant cycle of formation and breakdown. But while they exist, they create an enormous surface area where plant and fungus can exchange materials. The arbuscule might occupy a substantial portion of the cell's volume, a structure with dozens or hundreds of fine branches, all enclosed within the stretched and folded cell membrane. The fungus delivers nutrients through this membrane, passing phosphorus and nitrogen directly into the cell's interior. The plant packages sugars and passes them back to the fungus through the same membrane. The arbuscule is the heart of the partnership, the site where the ancient collaboration physically manifests at the cellular level, where the boundary between organism and organism becomes blurred, where two utterly different life forms achieve a level of integration that approaches a single unified system. But this is not merely a story of individual partnerships, of single fungi associating with single plants in isolated one-on-one relationships. The networks are far more complex, far more extensive, far more communal than a simple pairing. In a forest, a single tree may host dozens of different fungal species on its roots. A young seedling, only a few years old, might have 15 or 20 distinct mycorrhizal species colonizing different parts of its root system, each fungus bringing slightly different capabilities, slightly different nutrient gathering strategies. A mature tree in an old-growth forest can have over 50 different fungal partners, and some studies have found trees hosting more than 100 different species. Each fungus has its own characteristics, its own particular abilities to access certain nutrients, to tolerate certain soil conditions, to thrive in certain seasons. Some fungi are better at accessing phosphorus. Others excel at nitrogen. Some tolerate acidic soils. Others prefer alkaline conditions. Some grow actively in spring, others in fall. By hosting this diversity of partners, the tree ensures that it has access to nutrients under a wide range of conditions, that it is not dependent on the performance of any single fungal species. And these fungi, crucially, do not confine themselves to a single plant. Their hyphae spread through the soil, growing outward from one plant's roots, exploring, searching for nutrients, and inevitably they encounter the roots of other plants. And when they do, they often form mycorrhizae with those plants as well. Many fungal species are generalists, capable of forming partnerships with multiple plant species. This creates what researchers call a common mycorrhizal network. Tree. A hosts fungus X, which also colonizes tree B. Tree B hosts fungus Y, which also colonizes tree C. Tree C hosts fungus Z, which colonizes tree A. And on the web extends, threads connecting to threads, linking the forest into a biological network of staggering complexity. In a mature forest, nearly every plant is connected to nearly every other plant, not directly, but through one or two or three fungal intermediaries. The forest becomes a superorganism in a sense, a community linked by its underground connections into a system where resources and signals can flow from any plant to any other plant through the fungal network. Through this network, resources can move between plants, traveling from individual to individual in patterns that seem almost impossible, almost too cooperative to believe. Carbon fixed by one tree through photosynthesis can travel through fungal hyphae and appear in another tree entirely. Sometimes a tree of a different species, sometimes a tree meters, or even tens of meters away. This is not speculation or hypothesis. It has been measured repeatedly using radioactive carbon tracers. Experiments where researchers label the carbon dioxide around a tree's leaves, wait for the tree to photosynthesize, and incorporate that carbon into sugars. Then track where the radioactive carbon appears. In forests in British Columbia, in careful experiments conducted over multiple years, researchers labeled paper birch trees with radioactive carbon dioxide. Within days, that radioactive carbon appeared in nearby Douglas fir trees, having traveled underground through the shared mycorrhizal network. The transfer was significant, sometimes amounting to several % of the receiver tree's total carbon. Enough to measure, enough to matter for the tree's growth and survival. And the transfer was not one way. When Douglas firs were labeled, carbon appeared in birch trees. The flow was bidirectional, and crucially, the direction and magnitude depended on the time of year, on the season, on which tree had more photosynthate to spare, and which was under greater stress or need. In spring, when birch leaves emerge before Douglas fir needles, when the birch is photosynthesizing and the fir is not, carbon flows from birch to fir. The birch supports the fir during the fir's vulnerable period before its needles have fully developed. In late summer and fall, when birch growth slows and begins preparing for winter, while Douglas fir is still photosynthesizing strongly, carbon flows from fir to birch. The system self-regulates, resources flowing toward need, the network balancing supply and demand across the community, like a biological economy where wealth redistributes automatically toward those who need it most. When researchers deliberately shaded paper birch trees, blocking most of their light and reducing their photosynthesis, carbon transfer from connected Douglas fir trees increased by up to 10%, a dramatic demonstration that the network responds to need. That stressed plants can draw support from healthier neighbors through the fungal connections. This network does not merely transport carbon. Signals travel through it as well. Information transmitted from plant to plant through the threads, warning signals and chemical messages that allow plants to respond to threats they have not directly experienced. When a Douglas fir tree is attacked by insects, when beetles bore through its bark or caterpillars begin eating its needles, the tree responds by producing defensive compounds, chemicals that taste bad or are toxic to the attackers, and it increases production of volatile chemicals that attract predatory insects, calling in the cavalry to attack the herbivores. But something else happens. Something that seems almost like intelligence, like forethought. Nearby ponderosa pines, connected to the injured Douglas fir through the mycorrhizal network, begin producing defensive enzymes, even though they have not been attacked, even though no insects have touched them. The signal has traveled through the fungal threads. A chemical alarm transmitted underground that causes uninjured trees to mount defenses preemptively, to prepare for an attack that might never come but might be imminent. This is communication, or something functionally equivalent to it, transmitted through the underground network. The trees do not have nervous systems, do not have brains, cannot think in any sense we would recognize. Yet they participate in a system of information exchange that allows them to respond collectively to threats, to share information about danger, to prepare defenses before the danger reaches them. The implications extend even further, into territory that seems almost eerie in its suggestion of plant awareness. Trees appear capable of recognizing relatives, of distinguishing kin from strangers, of directing resources preferentially toward genetic relatives. Douglas fir seedlings grown in proximity to genetically related individuals siblings or close kin that germinated from seeds produced by the same parent tree, show greater carbon transfer through the network than when grown with unrelated individuals of the same species. The kin seedlings receive more support. They grow better, survive at higher rates. The mechanism remains unclear. We do not know how a tree recognizes relatives, what signals or chemical markers allow it to distinguish siblings from strangers. But somehow, the network facilitates kin recognition, allowing trees to favor relatives, to invest resources more heavily in the success of their genetic kin. This suggests a level of complexity in the network we are only beginning to unravel. This suggests information, opportunities that emerge from the sum of individual actions. The network's architecture itself reveals additional layers of complexity, patterns that suggest the system has evolved not just functional efficiency, but structural organization reminiscent of designed communication networks. In detailed mapping studies of old-growth Douglas fir forests, researchers painstakingly traced the mycorrhizal connections between trees, identifying which fungal species connected which trees, building maps of the underground network. What they found was surprising. The network is not uniformly distributed. It is not a random web where every tree connects to every other tree with equal probability. Instead, it has structure, organization, hierarchy even. Certain large, old trees function as hubs, highly connected nodes with mycorrhizal links to dozens or even hundreds of other trees. These hub trees, sometimes called mother trees, maintain connections to many younger trees, serving as central nodes in the network. When you map the connections, you see a pattern familiar from other types of networks, a pattern seen in airline route maps where certain cities serve as major hubs, in the internet where certain servers handle vastly more traffic than others, in social networks where certain individuals know far more people than average. The mycorrhizal network displays this same hub structure, with certain trees occupying central positions in the web of connections. These hub trees are typically the oldest, largest trees in the forest. They have had centuries to establish connections, to partner with many fungal species, to grow extensive root systems that explore vast volumes of soil. Their roots are colonized by fungal networks that spread for tens of meters in all directions, encountering and connecting with the roots of dozens or hundreds of other trees. A single large Douglas fir might be directly connected through shared fungi to 200 smaller trees within its vicinity. And because these large trees photosynthesize at high rates, producing enormous quantities of sugar thanks to their vast canopy of needles, they can afford to support other trees through the network, to subsidize seedlings that are struggling in shade, to maintain connections that may seem one-sided, but that ultimately benefit the forest as a whole. When researchers experimentally removed hub trees from forest plots, the results were dramatic. The network fragmented. Connectivity between remaining trees decreased sharply, and crucially, seedling survival declined. The young trees, no longer connected to the large hubs, struggled more, died at higher rates. The hub trees were not merely large and old, not merely taking up space and resources. They were integral to the network's function, serving as conduits through which resources and signals flowed, facilitating the establishment and growth of younger trees. stabilizing the community. There is even evidence, though it is harder to measure and more controversial, that before a mature tree dies, whether from age or disease or injury, it transfers resources into the network, making them available to younger trees in a final act of contribution. The tree, sensing perhaps through biochemical changes that its time is ending, appears to push stored nutrients into the fungal network rather than investing them in its own maintenance or reproduction. It is a nutrient legacy, a final gift bequeathed through the fungal network to the next generation. The carbon and nitrogen and phosphorus that the tree accumulated over its lifetime, that it pulled from the air and soil over centuries, does not simply decompose slowly after death. Some of it is actively transferred to living trees, while the dying tree's roots are still functional, still connected to the network. This has not been definitively proven, remains an area of active research, but the preliminary evidence suggests it happens, that dying trees support the growth of their successors, that the network facilitates a kind of intergenerational transfer of resources from old to young. This has profound implications for forest management, for how humans interact with and harvest forests. The common practice in commercial forestry is to remove the largest, most economically valuable trees, the trees with straight trunks and minimal branches, the trees that will yield the most valuable lumber. These are precisely the trees most likely to serve as network hubs. By selectively removing them, by high-grading the forest for the best individuals, we may be disrupting the subterranean connections in ways that impair forest regeneration and resilience in ways we have not fully appreciated. The remaining trees, even if they survive the logging operation, are suddenly less connected, less able to exchange resources and signals, less buffered against stress. Seedlings establishing after the harvest have fewer hub trees to connect with, fewer sources of subsidy during their vulnerable early years. The entire community may become less stable, less able to respond to drought or disease or insect outbreaks. There is growing recognition among foresters and conservation biologists that maintaining connectivity, leaving some large old trees, even in managed forests, preserving the structure of the underground network, may be as important for long-term forest health as any other management consideration. The mycorrhizal network influences forest dynamics at multiple scales, from the fate of individual seedlings to the structure of entire communities. At the finest scale, that of individual seedlings, connection to the network can be the difference between life and death. In the dense shade of a forest understory, where perhaps only 1 or 2% of the sunlight reaching the canopy makes it to the ground, photosynthesis is severely constrained. A seedling germinating in such conditions faces a nearly impossible challenge. It must produce enough sugar through photosynthesis to build roots, to build leaves, to power its metabolism, to grow, all while receiving barely enough light to drive photosynthesis at all. Many seedlings fail. They germinate, grow for a few weeks on energy stored in the seed, then die once those reserves are exhausted, unable to photosynthesize fast enough to survive. But seedlings that connect to the mycorrhizal network have another option. They can receive carbon from the network, subsidized by mature trees overhead that have full access to sunlight. The mature trees, photosynthesizing vigorously in full sun, produce far more sugar than they need. Some of that excess flows into the fungal network, and some of it reaches the struggling seedlings below. It is a subsidy, a transfer of resources from those who have plenty to those who have barely enough. With that support, seedlings can persist in deep shade for years, decades even, growing slowly, sometimes barely growing at all, but surviving, waiting for their chance. When a mature tree falls, when disease or wind or age creates a gap in the canopy, suddenly light floods down to the forest floor. The seedlings that have been waiting, that have been surviving on network subsidies, suddenly have access to the light they need. They accelerate their growth, race upward to fill the gap, and because they already have established root systems, already have height and structure, they can often outcompete new seedlings germinating in the gap. Without the network, this strategy would not work. The understory seedlings would die long before any gap formed. The network allows them to wait, to persist, to be ready. At larger scales, the network affects which species can coexist, how communities assemble, how ecosystems respond to disturbance and change. Because different fungal species have different ecological preferences and different nutrient acquisition capabilities, a diverse mycorrhizal community allows the plant community to access a wider range of resources, to exploit the full heterogeneity of the soil environment. Some fungi are better at accessing phosphorus bound to organic matter. Others excel at taking up inorganic phosphorus. Some can access nitrogen from decomposing organic material. Others work primarily with mineral nitrogen. Some tolerate low pH. Others prefer neutral or alkaline conditions. Some grow actively in spring when soils are cool and wet. Others peak in late summer when soils are warm and dry. By hosting this diversity of partners, by maintaining connections to multiple fungal species with different capabilities and different timing, plants ensure they have access to nutrients under a wide range of conditions. They are not dependent on the performance of any single fungal species, not vulnerable to the specific requirements or limitations of one partner. This resource complementarity, this functional diversity in the mycorrhizal community, stabilizes the system, buffering it against environmental variation. In wet years, when water is abundant, but nitrogen may be scarce, because wet soils slow decomposition, one suite of fungi dominates. Fungi good at accessing nitrogen from resistant organic compounds. In dry years, when nitrogen mineralization accelerates, but phosphorus becomes less mobile in dry soil, a different suite becomes more important. Fungi with strategies for accessing bound phosphorus. The network allows the forest to adjust, to respond flexibly to the shifting mosaic of conditions, to maintain productivity and diversity across years of varying weather and soil chemistry. These mycorrhizal associations, this ancient partnership between plants and fungi, are not limited to wild ecosystems, not confined to forests and prairies and tundra, where humans have not yet imposed their will. Agriculture depends on them as well. Or at least it should. Though modern farming practices have often disrupted these relationships in ways that may have seemed necessary at the time, but now appear short-sighted. In natural systems, when land is left fallow, allowed to rest between periods of cultivation, mycorrhizal networks remain intact in the roots of weeds and cover crops and any other plants that volunteer. The fungi persist, connected to living roots, continuing to exchange carbon for nutrients, maintaining the network's structure even though the crops are gone. When the next crop is planted, when seeds germinate and young roots push down into the soil, seedlings rapidly connect to the existing network. The fungi are already there, already established, and they quickly colonize the new roots, forming mycorrhizae within days. The crop gains immediate access to a vast foraging system, a network that may have been developing for years, that extends far beyond the young plant's own limited root system. The seedling can draw on the network from its earliest days, gaining nutrients more efficiently, establishing faster, growing more vigorously. But in intensive modern agriculture, these networks are repeatedly destroyed. Plowing turns the soil, breaking up the fungal hyphae, severing the connections, burying fungal structures deep where they cannot easily reconnect with roots. Deep tillage, the practice of turning soil to depths of 30 or 40 centimeters to break up compaction or incorporate amendments, is especially destructive, essentially resetting the fungal community each time it is performed. The application of broad-spectrum fungicides, used to control fungal diseases of crops, kills mycorrhizal fungi as well as pathogens, making no distinction between beneficial partners and harmful parasites. The practice of leaving fields bare between crops, either because of short rotations or because of weed control strategies, means there are no living roots for fungi to colonize. And without a host providing carbon, the fungi decline, their populations crash. When the next crop is planted, it must establish new partnerships from scratch, colonizing roots with whatever fungal spores happen to be present in the soil. And these spores may be scarce after the soil has been repeatedly disturbed and left plant-free. This slow recolonization is less efficient than connecting to an existing network. The crop's roots are colonized more slowly, less completely, and by a less diverse community of fungi, possibly by fungi that are not ideally suited to the crop or the soil conditions. The result is plants that are less vigorous, that access nutrients less efficiently, that are more dependent on added fertilizers to compensate for their reduced ability to forage for phosphorus and other nutrients through mycorrhizal networks. We have, in effect, disabled one of the plant's primary nutrient acquisition mechanisms, and then compensated by providing nutrients in more direct but often less sustainable forms. Through fertilizers that require energy to manufacture, that can run off into waterways causing pollution, that represent an ongoing cost rather than a self-sustaining biological system. There is growing interest among agronomists, soil scientists, and progressive farmers in practices that preserve or restore mycorrhizal networks in agricultural systems. Reduced tillage, where soil is disturbed minimally or not at all, allows networks to remain more intact, gives fungi a better chance to persist between crops. Cover cropping, where fields are planted with non-cash crops during fallow periods, specifically to maintain living roots in the soil, gives the fungi hosts to colonize, keeps the networks alive and active. Crop rotation that includes mycorrhizal dependent species, that ensures there are always plants in the rotation that host mycorrhizal fungi, maintains the fungal community's diversity and abundance. Some farmers are even experimenting with inoculation, adding mycorrhizal fungal spores to seeds or soil to boost colonization rates, especially in heavily degraded soils where native fungal populations are depleted. The goal in all these practices is the same: to maintain or restore the underground support systems, the ancient partnerships that have sustained plant life for hundreds of millions of years, allowing crops to benefit from the same biological infrastructure that supports wild plants in natural ecosystems. Let your mind rest on that image for a moment. Let yourself sink into the wonder of it. Below the surface of every garden, every field, every forest, every patch of ground where plants grow, invisible threads are weaving plants together, creating networks of exchange and communication that function continuously, without pause, day and night, summer and winter. Season after these networks are not static, not fixed structures that form once and remain unchanged. They are dynamic, living, constantly responding and adjusting, growing in response to need, retracting when resources are limited, connecting and disconnecting as plants come and go. As fungi spread and retreat, as the community shifts through time, adjusting to the needs and contributions of their participants, to the changing availability of water and nutrients, to the shifting balance of supply and demand across the network. When a plant dies, when its roots cease functioning and begin to decompose, the fungal network incorporates those nutrients, breaks down the organic matter, mineralizes the elements, and redistributes them to living plants. Nothing is wasted. The death of one organism becomes the sustenance of others, mediated through the fungal network. When a new seedling germinates, when a seed cracks open and a tiny root pushes down into the soil, fungal hyphae sense the presence of this new root, detect the chemical signals it releases, and begin the process of colonization. They grow toward it, contact it, begin the molecular negotiation that leads to mycorrhiza formation, extending the network to include this newcomer, welcoming it into the community of connected plants. The network is ancient. Its basic template, the fundamental mechanism of plant-fungus partnership, was laid down more than 400 million years ago, when plants first attempted to colonize land, when the Earth's surface was a rocky, inhospitable place where organic matter was scarce and nutrients were locked in, minerals that plants could not easily access alone. Yet the network is renewed constantly, perpetually refreshed. Each generation of plants and fungi enters into the partnership anew, finds the same mutual benefits their ancestors did, participates in the same collaborative system. The mechanism is ancient, but the participants are new. Each fungus engaging in a partnership that is at once exactly what countless This is a partnership that has sustained terrestrial life through ice ages and mass extinctions, through periods of global warming and cooling, through continental drift and mountain building, through asteroid impacts and volcanic eruptions, through every upheaval Earth has experienced since the Devonian period. It has adapted, evolved, diversified, but the core relationship has endured. Plants provide carbon. Fungi provide nutrients and water and connections. Both benefit. Both depend on the relationship. Neither could thrive. Perhaps neither could survive without the other. And this is a partnership that requires nothing from us, that functions without human awareness or intervention or approval. For most of human history, we did not know these networks existed. We saw forests and grasslands, crops and gardens, but we did not see the underground connections, did not understand that the plants were linked, that resources and signals were flowing between them through fungal intermediaries. The partnership operated in ignorance of our ignorance, followed its own logic, its own economy of exchange and reciprocity, functioned according to principles we are only now beginning to understand. It did not need our understanding to work. It does not need our permission to continue. And yet, it supports us, for we depend utterly on plants, on forests and crops and grasslands. We depend on them for food, for oxygen, for climate regulation, for the habitability of the planet. And those plants, in turn, depend on their fungal partners, on the quiet conversations happening in the soil beneath our feet, on the ancient partnership that makes terrestrial ecosystems possible. We are supported, ultimately, by relationships we cannot see, by partnerships that predate our species by hundreds of millions of years, by biological systems that function in darkness, in silence, beyond our direct perception, but that are as essential to life on land as sunlight or water or air. Beneath the surface of the soil, in a world as removed from our perception as the mycorrhizal networks, but equally essential to life, another ancient partnership operates, another collaboration between organisms so different they might seem to have nothing to offer each other. This partnership does not involve fungi. It involves bacteria, organisms so small that millions could fit on the head of a pin, yet capable of performing a chemical transformation that humans can replicate only with enormous inputs of energy, heat, and pressure in industrial facilities. These bacteria can take nitrogen from the air, inert nitrogen gas that makes up 79% of the atmosphere, but is utterly unusable by plants in that form, and transform it into ammonia, into a form of nitrogen plants can absorb and use. This transformation, this fixation of nitrogen, is one of the most important biological processes on Earth, fundamental to agriculture, to ecosystem productivity, to the protein content of the food we eat. And it happens through partnership, through symbiosis between bacteria and plants, primarily plants in the legume family, the family that includes beans and peas and clover and alfalfa and soybeans, crops that have been recognized for thousands of years as somehow special, somehow able to enrich soil rather than deplete it, somehow able to grow vigorously without the nitrogen fertilizers other crops require. The bacteria belong to a group collectively called rhizobia, a diverse collection of species within the alpha, proteobacteria, bacteria related more closely to certain plant pathogens than to each other, bacteria that acquired the genes for nitrogen fixation through horizontal gene transfer, through the passing of genetic material between unrelated organisms. They live in the soil as free, living individuals, surviving on whatever organic carbon they can find, dividing slowly, waiting. And they wait for a signal, a chemical signal released by legume roots, released specifically when the plant senses that nitrogen is scarce, that the soil does not contain enough nitrogen in available forms to meet the plant's needs. The plant roots secrete flavonoids, plant-produced compounds, into the rhizosphere, the soil immediately surrounding the root. These flavonoids serve multiple functions. They act as chemoattractants, drawing bacteria toward the root, but more importantly, they act as molecular switches, activating specific bacterial genes, the nod genes, nodulation genes. These genes, when activated, cause the bacteria to produce their own signal molecules, called nod factors. These nod factors are lipocheetolycosaccharides, complex molecules with a backbone of sugar units and a lipid tail, and they carry substitutions and modifications that determine host specificity, that determine which bacteria can partner with which plants. The specificity is remarkable. A rhizobia strain that can form a functional partnership with soybeans may be unable to nodulate beans. Each legume species, sometimes each legume variety, recognizes only certain nod factor structures, will respond only to bacteria producing the right molecular key. When a root encounters the right nod factor, when the plant's receptor proteins on root hair cells bind to the bacterial signal, a cascade of events begins. The root hair, that fine extension of the root epidermis, that increases surface area for water and nutrient absorption, begins to curl. It curves around, forming a structure called a shepherd's crook, and within that curl bacteria accumulate. The curling traps bacteria against the root hair surface, concentrating them, holding them in place, and then the bacteria begin to penetrate. They do not break through the cell wall by force. Instead, the plant itself facilitates their entry. The root hair cell membrane invaginates, folds inward, forming a tube-like structure called an infection thread. The bacteria enter this thread and begin dividing, multiplying within it, and the infection thread grows, extends deeper into the root hair, down through the epidermis, into the cortex of the root. Meanwhile, something else is happening, something that normally would not occur in a mature root. Cells in the root cortex, cells that have not divided in months or years, that have been differentiated and stable, suddenly begin dividing again. The nod factors trigger cell division in the cortex, creating a nodule primordium, the beginning of a new organ. This is extraordinary. The plant is creating a new structure, a new organ, specifically to house bacteria, dedicating resources and developmental machinery to constructing an environment where the bacteria can live and function. As the infection thread reaches the cortex, as it penetrates into the dividing cells of the developing nodule, the bacteria are released from the thread into the plant cells, but they are not released freely into the cytoplasm. Each bacterium becomes surrounded by a plant-derived membrane, the symbiosome membrane, creating a compartment within the cell. A membrane-bound space where the bacterium resides, and within this compartment, the bacterium undergoes a profound transformation. It changes shape, elongating, branching, sometimes becoming 4 or 5 times its normal size. Its DNA replicates without the cell dividing, leading to polyploidy, to cells with multiple copies of their genome. Its cell wall properties change. Its metabolism shifts dramatically. It becomes a bacteroid, a form specialized for nitrogen fixation, a form that in many legumes can no longer reproduce, can no longer return to a free-living state, has become committed irrevocably to the symbiotic relationship. This transformation is controlled by the plant. The plant produces hundreds of small peptides nodule, specific cysteine, rich peptides called NCR peptides in certain legumes, and these peptides, which have antimicrobial properties, essentially force the bacteria to differentiate, to commit to their nitrogen-fixing role, to become permanently dependent on the plant. The relationship becomes obligate for the bacterium. though the plant retains the option to sanction or terminate nodules that are not performing well, that are not fixing sufficient nitrogen. The mature nodule is a remarkable structure, an organ that did not exist on the root before infection, that grew specifically in response to bacterial signals, and that now houses millions of bacteroids within its cells. If you dig up a healthy legume plant midway through the growing season and wash the soil from its roots, you can see the nodules, small, round, or finger-shaped growths attached to the roots, ranging in size from a few millimeters to a centimeter or more, depending on the plant species and the age of the nodule. And if you slice one of these nodules open with a knife or razor blade, you see something remarkable. The interior is pink, sometimes deep pink, sometimes almost red, a color unlike anything else in the root. Unlike the white or pale yellow of normal root tissue, this color is caused by a protein called legemoglobin. Leg for legume, hemoglobin because it is structurally similar to the hemoglobin in your blood, the protein that carries oxygen to your tissues. Legemoglobin is a plant protein containing a heme group, an iron containing molecule that binds oxygen and turns red when oxygenated, and the plant produces this protein in enormous quantities within the nodule, concentrations reaching millimolar levels in the cells housing bacteroids. A fully functional nodule may contain legemoglobin at concentrations comparable to hemoglobin in blood, making the nodule interior look like muscle tissue, pink with oxygenated protein. But why does the nodule need so much hemoglobin? Because of a paradox at the heart of nitrogen fixation, a fundamental tension in the requirements of the process. Nitrogen fixation. Nitrogen gas to ammonia is performed by an enzyme called nitrogenase. This enzyme is the only biological catalyst capable of breaking the triple bond between the two nitrogen atoms in N2, a bond so strong that nitrogen gas is essentially inert under normal conditions. Breaking that bond requires enormous energy, supplied as ATP, and requires electrons, reducing power. The bacteroids must respire actively, must run their metabolism at high rates to generate the ATP needed for nitrogen fixation. And respiration requires oxygen. Oxygen is the terminal electron acceptor in aerobic respiration, the molecule that allows the bacteria to extract maximum energy from the carbon compounds the plant supplies. Without sufficient oxygen, the bacteroids cannot respire efficiently, cannot generate enough ATP, cannot sustain nitrogen fixation. But here is the problem, the paradox. Nitrogenase, the enzyme that actually fixes nitrogen, is extraordinarily sensitive to oxygen. Oxygen inactivates it, binds to metal centers in the enzyme, and destroys its catalytic activity. The enzyme works only in low oxygen or oxygen-free conditions, so the nodule faces an impossible requirement. It needs high oxygen to power respiration and ATP production, but it needs low oxygen to protect nitrogenase. The solution is legemoglobin. Legemoglobin acts as an oxygen buffer and transport system. It has a very high affinity for oxygen, roughly 20 times higher than myoglobin in your muscles, meaning it binds oxygen tightly, holds onto it. When legemoglobin is present at high concentrations, it binds most of the oxygen in the nodule, keeping the concentration of free oxygen, Oxygen dissolved in the cytoplasm, unbound to protein, extremely low. Free oxygen concentrations inactively nitrogen. Fixing nodules are kept in the nanomolar range, roughly 10 to 30 nanomolar, low enough that nitrogenase is not inactivated, but the total oxygen content, counting both free oxygen and oxygen bound to legemoglobin, is much higher. The legemoglobin serves as an oxygen reservoir and facilitates oxygen transport. It picks up oxygen from the nodule's outer tissues, where oxygen diffuses in from the air spaces in the soil, then releases it gradually to the bacteroid membranes, where it can be used for respiration. The system works because legemoglobin can bind and release oxygen rapidly, cycling between oxygenated and deoxygenated forms, maintaining a high flux of oxygen to the bacteroids for respiration, while keeping the free oxygen concentration low enough to protect nitrogenase. It is an elegant solution to a challenging problem, a finely tuned balance between opposing requirements. When you cut open a nodule and see pink interior tissue, you are seeing visual evidence of this balance. Seeing the oxygenated legemoglobin that indicates active nitrogen fixation is occurring. Nodules that are not actively fixing nitrogen look different. Young nodules, before nitrogen fixation starts, are often white or gray inside. They have not yet produced significant legemoglobin, have not yet established the oxygen management system needed for nitrogenase activity. Old nodules, past their productive phase, turn green as the heme breaks down and releases iron as the structures deteriorate. As the plant ceases supporting them and begins to dismantle them. And ineffective nodules, nodules formed by bacterial strains that are poor at nitrogen fixation, or nodules stressed by environmental conditions, also lack the characteristic pink color. They may be small, pale, unable to provide significant nitrogen to the plant. A farmer or agronomist evaluating whether a legume crop is fixing nitrogen can simply dig up a few plants, slice open nodules, and look at the color. Pink or red nodules, especially if they are numerous and of good size, indicate active, healthy nitrogen fixation. White, gray, or green nodules indicate problems, either ineffective bacteria, poor environmental conditions, or plants that are not investing in the symbiosis because soil nitrogen is already plentiful. The nitrogen fixation process itself is energetically expensive. The conversion of nitrogen gas to ammonia requires tremendous reducing power and ATP. The overall reaction can be summarized as nitrogen gas plus 8 electrons, plus 16 ATP plus 8 protons, yielding 2 molecules of ammonia, plus 1 molecule of hydrogen gas, plus 16 ADP plus 16 inorganic phosphate. For every molecule of nitrogen fixed, the bacteroid must invest 16 ATP molecules. This represents an enormous energy cost. The carbon compounds the bacteroid receives from the plant, primarily organic acids like malate and succinate, are metabolized through respiration to generate this ATP. Different legume species differ in their efficiency. Cowpea, for instance, requires about 3 milligrams of carbon to fix 1 milligram of nitrogen, a relatively efficient ratio. White lupin is less efficient, requiring over 6 milligrams of carbon per minute kilogram of nitrogen fixed. And even in efficient species, the plant may divert up to 50% of its photosynthate, of the sugars produced through photosynthesis to the nodules during peak nitrogen fixing periods. This is an enormous investment. Half of everything the plant produces through photosynthesis goes to feed bacteria. But the investment is worthwhile because the nitrogen the plant receives in return is the primary limiting nutrient in most soils, the nutrient most likely to constrain plant growth. With sufficient nitrogen, a plant can produce proteins, nucleic acids, chlorophyll, all the nitrogen-containing compounds essential to life. Without nitrogen, growth stops. The amount of nitrogen fixed varies by crop and conditions, but can be substantial. Grain legumes like soybeans typically fix 50 to 250 pounds of nitrogen per acre over a growing season. Some sources cite values up to 250 pounds per acre for productive soybean crops under good conditions. Perennial forage legumes like alfalfa and clover can fix even more, between 250 and 500 pounds of nitrogen per acre annually, with alfalfa particularly impressive in its nitrogen fixation capacity. These quantities are comparable to the nitrogen application rates in conventional agriculture, where non-legume crops might receive 100 to 200 pounds of nitrogen per acre as fertilizer. The legume, through its partnership with bacteria, obtains comparable nitrogen supplies without requiring synthetic fertilizer, without the energy inputs and environmental costs associated with industrial nitrogen fixation. And importantly, not all of the fixed nitrogen is removed when the crop is harvested. A significant portion remains in the root system, in nodules, in root tissue, and in crop residues left in the field. As these materials decompose, they release nitrogen into the soil, making it available to subsequent crops. This is why legumes have been valued in crop rotations for millennia. Why farmers planting corn or wheat after clover or alfalfa often see improved yields without adding extra nitrogen fertilizer. Why green manure crops, legumes grown specifically to be tilled into soil, can improve soil fertility. The legacy of the symbiosis extends beyond the legume itself, enriching the soil for future plants. But this system is not automatic, not guaranteed to work in every field or every situation. The rhizobia bacteria must be present in the soil, and they must be the right strains. Strains compatible with the legume being planted, and strains effective at fixing nitrogen. In soils where a particular legume has not been grown before, the native rhizobia population may be absent or very low. A soybean planted in a field that has never grown soybeans may not nodulate well, may struggle due to nitrogen deficiency, even though the capacity for nitrogen fixation exists in the plant's genes. The solution is inoculation, deliberately adding the appropriate rhizobia strains to the seed or soil before planting. Commercial inoculants are available, products containing billions of live bacteria in a carrier material, formulated to be applied to seed just before planting or mixed into the soil. The bacteria in these inoculants are selected strains that have been tested and found to be highly effective at nitrogen fixation with specific legume species. Using inoculants can dramatically improve nodulation and nitrogen fixation. But the bacteria are fragile. They are sensitive to heat, to desiccation, to UV radiation from sunlight. Inoculated seed, should be planted promptly, ideally within 24 hours, and should be drilled into the soil rather than broadcast on the surface, where the bacteria will be exposed to sunlight and heat. Even with proper inoculation, conditions must be favorable. The bacteria need moisture to survive and move to root hairs. They need moderate temperatures. Extreme heat or cold, drought, soil compaction, poor soil fertility, all can reduce nodulation success and nitrogen fixation rates. The relationship between legume and rhizobia is not always harmonious. There is inherent tension, potential for conflict, because the bacteria and plant do not always have aligned interests. From the plant's perspective, the ideal bacterium is one that fixes nitrogen at high rates, provides maximum benefit, and reproduces minimally, directing its resources toward nitrogen fixation rather than toward producing the next generation of bacteria. From the bacterium's perspective, the ideal situation might be to receive carbon from the plant while doing as little nitrogen fixation as necessary to avoid sanctions, reserving resources for reproduction. This creates the potential for cheating. For bacteria that take carbon without providing nitrogen, and indeed, researchers have found variation in nitrogen fixing efficiency among rhizobia strains. Some strains form nodules, but fix little or no nitrogen, essentially parasitizing the plant. Plants have evolved mechanisms to counter this, to impose sanctions on underperforming nodules. If a nodule is not fixing nitrogen efficiently, the plant can reduce the carbon supply to that nodule, can reduce oxygen flow, can trigger early senescence and nodule death. This sanctioning mechanism helps maintain cooperation, helps ensure that bacteria fixing more nitrogen receive more resources, creating selection pressure for mutualism over parasitism. The system works, has worked for millions of years, but it is not perfect. There is always variation, always some nodules performing better than others, always tension between the partners even as they cooperate toward shared goals. Shift now to a different ecosystem, to a different partnership, from the dark soil to sunlit tropical seas, from bacteria and roots to algae and animals. Imagine a coral reef, the most diverse ecosystem in the ocean, sometimes called the rainforest of the sea. The physical structure of the reef is built by corals, animals in the phylum Cnidaria, related to sea anemones and jellyfish. Each coral is actually a colony of many small polyps, individual animals just a few millimeters across, living together and secreting a shared calcium carbonate skeleton that accumulates over years and centuries into the massive limestone structures we recognize as coral reefs. The polyps have tentacles armed with stinging cells to capture zooplankton from the water, and they feed by catching and consuming these tiny drifting animals. But in most reef-building corals, in the corals that create the bulk of the reef structure, this feeding provides only a fraction of the nutrition the coral needs. The majority of the coral's energy comes not from hunting but from partnership, from symbiosis with microscopic algae living inside the coral's tissues. These algae belong to the dinoflagellate family Symbiodiniaceae, and they are commonly called zooxanthellae, a name meaning little yellow animals, though they are not animals at all but photosynthetic protists, single-celled organisms that photosynthesize like plants. Each coral polyp hosts hundreds of thousands to millions of these algal cells in its tissues. specifically in the gastrodermal cells lining the coral's gut cavity. The density can reach 1 to 2 million algal cells per second square centimeter of coral surface, a concentration so high that the algae give the coral its color. The brown or green or sometimes golden appearance of healthy coral is not the color of the animal tissue, but the color of the millions of algal cells packed within it. The algae photosynthesize, using sunlight penetrating the clear tropical water to fix carbon dioxide into sugars, and they transfer a large portion of these photosynthates to the coral host. Depending on conditions and species, the algae may transfer 60 to 90% of what they produce to the coral. This is an enormous subsidy. The coral receives the majority of its carbon nutrition from the algae, allowing it to grow and calcify far more rapidly than would be possible through feeding alone. Experiments comparing corals with zooxanthellae to corals without zooxanthellae show dramatic differences. Symbiotic corals can grow 10 times faster, calcify more readily, dominate shallow tropical reefs. Asymbiotic corals, corals without zooxanthellae, are restricted to deeper water or temperate regions, grow slowly, do not build reefs. In return for this carbon subsidy, the coral provides the algae with a protected environment, a stable home sheltered within animal tissue, and critically, it provides nutrients. The algae need nitrogen and phosphorus to grow, to build proteins and nucleic acids and membranes, but these nutrients are scarce in tropical waters. The clear blue water of coral reefs is clear precisely because it is nutrient poor, because it lacks the nitrogen and phosphorus that would support blooms of phytoplankton that would cloud the water. This seems paradoxical. Coral reefs are among the most productive ecosystems on Earth, yet they thrive in waters that are essentially marine deserts, waters with very low concentrations of dissolved nutrients. The solution is the symbiosis. The coral, through its feeding, captures zooplankton and other small organisms. It digests these prey, breaking down their proteins and nucleic acids, and in the process releases nitrogen and phosphorus as metabolic waste, as ammonia and phosphate. These waste products, instead of being excreted into the water where they would diffuse away, are retained and made available to the zooxanthellae. The algae absorb the nitrogen and phosphorus the coral produces, use these nutrients for growth, and in turn, photosynthesize faster, providing more carbon back to the coral. The nutrient cycle between the partners, retained within the symbiosis rather than lost to the surrounding water. This tight nutrient cycling allows coral reefs to be highly productive even in nutrient poor waters, allows them to support the immense biodiversity for which they are famous. The symbiosis is the foundation, the mechanism that makes the coral reef ecosystem possible. But this partnership, like all the partnerships we have explored, has vulnerabilities, points of fragility where stress can cause the system to break down. The coral-zooxanthellae symbiosis is particularly sensitive to temperature. The algae have an optimal temperature range, typically in the mid to high 20s Celsius, and they function well within that range, photosynthesizing efficiently, transferring carbon to the host, causing no problems. But when water temperature rises above normal, by even a degree or 2 Celsius, sustained for days or weeks, the algae become stressed. Their photosynthetic machinery begins to malfunction. They produce reactive oxygen species, harmful molecules that damage cells. Instead of being beneficial partners, the stressed algae become a liability. producing toxins that harm the coral. The coral responds by expelling the algae, ejecting them from its tissues. This is coral bleaching. The coral loses its algal symbionts, and without the colored algae, the white calcium carbonate skeleton shows through the translucent coral tissue, making the coral appear bleached, white or pale. A bleached coral is not dead, not yet, but it is in crisis. Without its algal partners, it has lost its primary energy source. It can still feed on zooplankton, but this provides only a small fraction of its needs. The coral begins to starve, to draw down its energy reserves, to reduce growth and reproduction to bare maintenance. If temperatures return to normal quickly, within a few weeks, the coral can reacquire zooxanthellae, either from remnant populations that remained in the tissues or from free-living zooxanthellae in the water, and the coral can recover. But if the thermal stress persists, if high temperatures last for months, the coral's reserves are exhausted. It begins to die, the tissues breaking down, the skeleton becoming overgrown with algae and other organisms. A single severe bleaching event can kill a large percentage of corals on a reef. Repeated bleaching events over years can devastate reef systems, transforming vibrant coral, dominated ecosystems into rubble fields overgrown with algae. Ecosystems that have lost their complexity, their productivity, their ability to support the fish and invertebrates that depend on them. And bleaching events are becoming more frequent. Ocean temperatures have risen roughly 1 degree Celsius over the past century, and they are projected to rise at least another degree, possibly more, over the coming decades. Marine heat waves, periods of abnormally high ocean temperature, lasting weeks or months, are becoming more common. What once might have been a once-per-decade event in a particular region is now occurring every few years, sometimes every year. Corals do not have time to recover fully between bleaching events. Reefs around the world are declining, coral cover decreasing, reef structure degrading. The symbiosis, so successful for millions of years, is being disrupted by temperature increases caused by human activity, by the accumulation of greenhouse gases in the atmosphere. The threshold for bleaching varies somewhat by coral species and by the zooxanthellae strain the coral hosts. But in general, sustained temperature increases of 1 to 2 degrees Celsius above the normal maximum for a region are sufficient to trigger bleaching. Some coral populations show signs of adaptation or acclimatization, developing tolerance to slightly higher temperatures. But the rate of temperature increase is outpacing the rate at which corals can adapt. The future of coral reefs is uncertain, contingent on how much additional warming occurs, on whether warming can be limited and stabilized, on whether corals can evolve heat tolerance fast enough to keep pace with changing conditions. One potential mechanism for adaptation involves the diversity of zooxanthellae. The family Symbiotidaceae includes many species and genetic variants, and different types have different temperature tolerances, different photosynthetic efficiencies, different growth rates. Corals can host different zooxanthellae types, and some corals show flexibility, the ability to change their dominant zooxanthellae type in response to environmental conditions. A coral might host primarily a temperature-sensitive type under normal conditions, benefiting from its high photosynthetic rate. But after bleaching, when recolonizing with zooxanthellae, might acquire a more heat-tolerant type. This partner switching could provide corals with enhanced resilience, allowing them to adjust to warming waters. Studies have shown that corals hosting more thermally tolerant zooxanthellae types can withstand temperatures about 1 to 1.5 degrees Celsius, higher than corals hosting less tolerant types before bleaching. This is significant. A 1-degree increase in bleaching threshold could buy corals crucial time, could be the difference between survival and extinction for some populations. But there are trade-offs. The heat-tolerant zooxanthellae types are often less productive under normal conditions, less efficient at photosynthesis, less generous with carbon transfer. Corals hosting them may grow more slowly, may be outcompeted by corals hosting the faster-growing but temperature-sensitive types. The optimal strategy likely depends on the environment, on how frequently thermal stress occurs, on whether faster growth or stress tolerance is more important for survival and reproduction in a particular location. And the rate and extent of partner switching in wild corals remains an area of active research. It is not yet clear whether this mechanism can allow corals to keep pace with rapidly warming oceans, whether the flexibility is sufficient to prevent widespread reef collapse. The coral-zooxanthellae symbiosis also raises fascinating questions about the evolution and maintenance of cooperation, about why the algae transfer so much of their photosynthate to the host rather than retaining it for their own growth and reproduction. From an evolutionary perspective, this seems altruistic, costly to the algae. They produce sugars through photosynthesis, then give most of it away to the coral rather than using it to divide and increase their own population. Why? The answer likely involves several factors. First, the coral provides resources the algae need, particularly nitrogen and phosphorus, which are limiting in the reef environment. By transferring carbon to the coral, the algae ensure the coral continues feeding, continues providing nutrients. It is an exchange, carbon for nutrients, though the exchange is mediated not through direct negotiation, but through evolutionary processes that have shaped the physiology of both partners. Second, the coral actively controls the algae population. It digests excess algae, maintains the population at a density that maximizes the coral's benefit, rather than the algae's. The algae are, in a sense, domesticated, maintained at high density, but prevented from dividing freely. Third, in some coral species, algae are passed from parent coral to offspring and eggs, a process called vertical transmission. In these species, the algae benefit from the coral's reproductive success, because successful coral reproduction is the algae's path to the next generation. This aligns the interests of algae and coral, makes investments in coral survival and growth investments in the algae's own genetic future. In other coral species, each new coral larva must acquire zooxanthellae from the environment, a process called horizontal transmission. Here, the alignment of interests is less perfect. More opportunities exist for conflict, but the coral's control over the algae population likely maintains cooperation even without direct genetic linkage across generations. Let your mind rest now on these two systems. On the legume nodule with its pink nitrogen, fixing interior, and the coral polyp with its golden zooxanthellae glowing in the sunlight. Both are partnerships between very different organisms, between autotrophs that can photosynthesize and heterotrophs that cannot. Both involve one partner providing carbon-based energy and the other providing nutrients or waste processing. Both have evolved elaborate mechanisms to facilitate the partnership. From the legume's nodule construction and legumoglobin production to the coral's precise regulation of zooxanthellae density and nutrient retention. Both partnerships are ancient, dating back millions of years, representing successful solutions to fundamental biological challenges. And both partnerships face modern threats, from changing climate to human modifications of ecosystems. The nitrogen-fixing symbiosis in agriculture is disrupted by tillage and fungicides and practices that break continuity of living roots. The coral-zooxanthellae symbiosis is threatened by warming oceans and bleaching events that exceed the partnership's tolerance limits. These are not merely abstract concerns for distant organisms. These partnerships support human well-being. Legume nitrogen fixation feeds billions of people, provides protein in diets around the world, enriches agricultural soils, reduces dependence on energy-intensive nitrogen fertilizers. Coral reefs support fisheries that feed hundreds of millions of people, protect coastlines from storms and erosion, support tourism economies, harbor biodiversity that may hold medical and scientific value yet to be discovered. The health of these partnerships matters, not just to the organisms directly involved, but to human societies that depend on the ecosystem services these symbioses provide, and understanding them, appreciating the elegance and complexity of these ancient collaborations, may help us protect them, may inspire approaches to agriculture and conservation that work with these partnerships rather than against them, that recognize the fundamental role of symbiosis in shaping and sustaining the living world. The chemical details of these exchanges reveal remarkable sophistication. In the legume nodule, the bacteroid receives malate and succinate from the plant, 4 carbon organic acids that serve as both carbon and energy sources. These acids are metabolized through the tricarboxylic acid cycle, generating electrons that flow through the respiratory chain, creating the proton gradient that drives ATP synthesis. The bacteroid uses this ATP to power nitrogenase, which catalyzes one of the most demanding reactions in biology. The enzyme contains iron and molybdenum at its active site, and it uses ATP hydrolysis to drive electrons onto the nitrogen molecule, progressively reducing it through intermediates until ammonia forms. The process requires 8 electrons and 16 ATP molecules per nitrogen molecule, an enormous investment that explains why nitrogen fixation is so energetically expensive. The ammonia produced is immediately converted by the plant into glutamine and asparagine, amino acids that serve as transport forms for nitrogen, carrying it from the nodule to the rest of the plant, where it is incorporated into proteins, nucleic acids, and other essential molecules. This entire metabolic integration requires coordination between plant and bacterial biochemistry, requires transport proteins to move organic acids into the symbiosome and to export amino acids out, requires signaling systems to match bacterial metabolism with plant needs, requires regulatory networks that respond to the plant's nitrogen status and adjust fixation rates accordingly. In the coral-zooxanthellae system, the photosynthates transferred from algae to coral include glucose, glycerol, and other small organic molecules. These pass from the algae through the symbiosome membrane into the coral cells, where they are metabolized to provide energy for growth, calcification, and maintenance. The process is not passive diffusion, but involves active regulation by both partners. The coral can influence how much carbon the algae transfer through mechanisms that are still being elucidated, possibly involving changes in the chemical environment around the algae, signals that affect algal membrane permeability or metabolic partitioning. The nitrogen and phosphorus flowing from coral to algae include ammonia from protein catabolism, urea from metabolism of nucleic acids, and phosphate from various cellular processes. These nutrients are limiting factors for algal growth. And by controlling nutrient supply, the coral essentially controls the algae population, keeping it at densities that maximize benefit to the coral, rather than allowing the algae to grow unchecked. This control is crucial because if zooxanthellae populations grew too large, they would compete with the coral for resources, would shade each other, reducing photosynthetic efficiency, could even physically damage coral tissues. The system maintains balance through multiple feedback loops, regulatory mechanisms, operating at cellular and molecular levels to keep the partnership stable and productive. These partnerships also teach us about the nature of cooperation itself, about how organisms with different interests can evolve to work together, can create relationships where both benefit even though neither is altruistic, neither acting for the other's good, but rather pursuing its own survival and reproduction in ways that happen to benefit the partner. This is the essence of mutualism. Not cooperation born of kinship or conscious choice, but cooperation that emerges because natural selection favors it. Because individuals that participate in beneficial partnerships leave more descendants than those that do not. The legume benefits from nitrogen fixation, but invests heavily in supporting the bacteria. The bacteria benefit from the plant's carbon, but expend enormous energy fixing nitrogen. Both investments are costly. Both reduce resources available for other purposes. Yet both pay off because the benefits exceed the costs. The coral benefits from algal photosynthesis, but provides nutrients and protection. The algae benefit from coral waste products and stable habitat, but transfer most of their production to the host. Again, costs and benefits balance in a way that makes the partnership more successful than either partner would be alone. Understanding this, recognizing that cooperation does not require consciousness or intent, but can emerge from purely selective processes, helps us understand the prevalence of symbiosis in nature, helps us see that partnerships are not rare exceptions, but fundamental patterns in the organization of life. From cells containing mitochondria and chloroplasts, organelles that were once free, living bacteria, to the complex multicellular organisms, built through cooperation between cells, to the relationships between species we have been exploring. Symbiosis operates at every scale, at every level of biological organization, shaping the diversity and complexity of the living world in ways we are only beginning to fully appreciate. Consider now a different kind of partnership, one that creates not just a relationship between organisms, but an entirely new form of life. A composite organism so integrated that for centuries naturalists classified it as a single species, unaware that what they were observing was actually multiple organisms living as one. These are lichens, those crusts and rosettes and hanging strands you see on tree bark and rocks and fence posts, organisms so ubiquitous, so adaptable, that they thrive from the Arctic to the Antarctic, from desert rocks to tropical rainforest canopies, from sea level to the highest mountains. conditions that would kill most other life forms. There are approximately 20,000 known species of lichens, though that number continues to grow as taxonomists describe new forms, and as molecular techniques reveal that what appeared to be a single lichen species is actually several cryptic species, visually indistinguishable but genetically distinct. And every single one of these 20,000 species is not a species in the traditional sense, but a partnership, a symbiosis between a fungus and one or more photosynthetic partners, typically either green algae or cyanobacteria, and sometimes both, and increasingly, we are learning that lichens also contain yeasts and bacteria as integral components, making them not partnerships between two organisms but communities, miniature ecosystems, complex assemblages of multiple species living together in intimate association, structured and organized in ways that create properties none of the partners possess alone. The dominant partner, the one that gives the lichen its structure and its species name, is always a fungus. About 98% of lichen forming fungi are Ascomycetes, the group that includes cup fungi and many other familiar forms, and the remaining 2% are Basidiomycetes, the group that includes mushrooms, but these are not random fungi, not species that happen to form lichens occasionally. About 20% of all fungal species have adopted the lichen lifestyle, have evolved specifically to form these associations, have become so specialized for symbiosis that most of them cannot survive alone. If you isolate a lichen fungus and grow it in the laboratory without its photosynthetic partner, it forms an undifferentiated mass of fungal filaments, a shapeless tangle of hyphae with no structure, no organization. But combine that same fungus with the appropriate algae or cyanobacteria, provide the right conditions, and a transformation occurs. The fungus begins to organize, to differentiate, to form the characteristic lichen structure. This process is called morphogenesis, the generation of form, and it demonstrates that the lichen structure, the thallus, emerges from the interaction between the partners, arises from their partnership rather than from either organism alone. The form of the lichen is a product of symbiosis, a structure that exists only when the partners are together. The structure itself is remarkable, a multi-layered construction where each layer has specific functions and houses specific organisms. In most lichens, the structure is heteromerous, meaning the fungal and photosynthetic components are arranged in distinct layers rather than mixed uniformly. The uppermost layer is the upper cortex, formed entirely by the fungus, consisting of tightly packed fungal hyphae, forming a dense protective layer. This cortex shields the lichen from excessive light, from desiccation, from physical damage, from many of the environmental stresses that would harm the more delicate photosynthetic cells within. Below the cortex lies the algal layer, where the photosynthetic partner resides. Cells of green algae or cyanobacteria, interspersed among fungal hyphae, positioned to receive light that penetrates the cortex but protected from the full intensity of direct sunlight. Below the algal layer is the medulla, a looser layer of fungal filaments with air spaces between them, serving functions of gas exchange and water storage, providing structural support without excessive weight. And at the base, in many lichens, is a lower cortex, another protective layer. And from this lower surface extend rhizines, thread-like structures that anchor the lichen to its substrate, to bark or rock or soil, holding it in place against wind and rain. The photosynthetic partners, the photobionts, are surprisingly limited in diversity given the 20,000 lichen species. Only about 100 species of algae and cyanobacteria are commonly found in lichens. About 90% of lichens associate with green algae, and within that group, one genus dominates. Trebuchet, a single-celled green alga, serves as the photobiont in about 20% of all lichens, an extraordinary degree of reliance on a single algal genus. Other green algal genera, like Trentipolia, also participate in lichen symbioses. But the diversity is limited, far more limited than the diversity of lichen-forming fungi. About 10% of lichens, the cyanolichens, associate with cyanobacteria instead of or in addition to green algae, most commonly with filamentous cyanobacteria in the genus Nostoc. And some lichens are tripartite, containing both a green algal photobiont distributed throughout the main body of the lichen and cyanobacterial cells confined to specialized structures called cephalodia, small gall, like growths where nitrogen fixation occurs. These tripartite lichens gain both carbon through photosynthesis from the green algae and nitrogen through fixation by the cyanobacteria, a double subsidy that allows them to thrive in extremely nutrient-poor environments. The exchange between fungus and photobiont follows a pattern we have seen in other symbioses. The photobiont photosynthesizes, fixing carbon dioxide into organic compounds, producing sugars. Different photobionts produce different specific compounds. Green algae typically produce polyols, sugar alcohols like ribitol or erythritol, while cyanobacteria produce glucose. These compounds are transferred from the photobiont to the fungus, and the transfer is not merely leakage or passive diffusion. The fungus actively manipulates the photobiont to increase transfer rates. The fungal hyphae in contact with photobiont cells can induce the photobiont to release or leak photosynthetic products at rates far higher than the photobiont would normally release. The fungus may produce compounds that affect photobiont membrane permeability, or may create chemical gradients that draw compounds out of the photobiont cells. The relationship, from the photobiont's perspective, may be less mutualism than controlled parasitism. The fungus farming the photobiont, maintaining it and protecting it, but also extracting a large fraction of its production. The fungus can capture a very high proportion of the carbon the photobiont fixes, immediately assimilating it and transforming it into compounds the photobiont cannot use, ensuring the carbon flows toward the fungus rather than being retained for photobiont growth. In return, the fungus provides the photobiont with water, mineral nutrients absorbed from rain and from the substrate, and protection from environmental extremes. The cortex shields the photobiont from excessive UV radiation, from desiccation during dry periods, from temperature fluctuations. The fungal structure provides a stable microenvironment where the photobiont can function even when external conditions would be lethal. Lichens can survive complete desiccation, losing virtually all their water content, ceasing all metabolic activity, and then resuming normal function within minutes or hours of being re-wetted. The photobiont cells, protected within the fungal matrix, can tolerate this desiccation, rehydration cycling indefinitely. Lichens have been frozen to temperatures approaching absolute zero and revived. They have survived years in vacuum and intense radiation in space, mounted on the exterior of spacecraft, exposed to conditions no other complex life form can tolerate. The protection the fungus provides makes this extreme tolerance possible, allows the lichen to colonize environments where neither partner could survive alone. The resulting lichen has properties neither partner possesses independently. Lichens grow slowly, often only a few millimeters per year, but they can live for extraordinary lengths of time. Some crustose lichens growing on Arctic rocks are estimated to be over 8,000 years old, possibly approaching 10,000 years, making them among the oldest living organisms on Earth, older than the oldest trees, older than any known animal. They grow so slowly that researchers can use them for dating rock surfaces, a technique called lichenometry, measuring lichen sizes to estimate how long a surface has been exposed. Lichens produce unique chemical compounds, secondary metabolites, that neither the fungus nor the photobiont produces in isolation. These lichen substances, of which hundreds are known, serve various functions. Some are pigments that absorb harmful UV radiation, protecting the photobiont. Some have antimicrobial properties, defending against bacteria or other fungi that might compete with or parasitize the lichen. Some may help regulate water relations or mediate interactions with the substrate. These compounds are so distinctive that lichens have been used for centuries as sources of dyes and medicines, and they remain of interest to pharmaceutical researchers seeking novel bioactive compounds. Lichens come in 3 main growth forms, each adapted to different environmental conditions. Crustose lichens form thin crusts adhering tightly to the substrate, growing into the surface of rock or bark, essentially inseparable from it, without destroying the lichen. These are the most drought-resistant form, the most common in extreme environments, like deserts or high mountains, places where water is scarce and conditions are harsh. Foliose lichens are leaf-like, forming lobed structures attached to the substrate at specific points, but with much of the thallus free, rising slightly above the surface. They are intermediate in their environmental tolerances and common in temperate forests on tree bark and rocks. Fruticose lichens are shrubby or hair-like branching structures that may hang from branches, like the old man's beard lichens of wet forests, or stand upright on soil or rock like miniature shrubs. These are the most susceptible to desiccation, the most restricted to areas where moisture is more reliably available, but they can achieve sizes and forms impossible for crustose types. Lichens reproduce through multiple strategies, some involving sexual reproduction of the fungus, others involving asexual dispersal of both partners together. The fungus can produce fruiting bodies, apothecia in Ascomycetes, that generate spores through sexual reproduction. These spores disperse and germinate, but the resulting fungal hyphae must then find compatible photobionts to reform the lichen, a process called lichenization that can take months or years and has uncertain success rates. Alternatively, many lichens produce specialized propagules that contain both fungal and photobiont cells together, allowing both partners to disperse as a unit. Soradia are small clusters of photobiont cells, wrapped in fungal hyphae, breaking off from the lichen surface and dispersing on wind or water, or carried by animals, Isidia are small projections from the lichen surface, containing both partners in proper proportions, breaking off and establishing new lichens. These vegetative propagules ensure that both partners remain together, that the successful partnership is maintained across generations, rather than requiring each fungal spore to find and establish symbiosis with photobionts anew. The ecological roles of lichens are profound. Despite their small size and slow growth, they are pioneer organisms among the first life forms to colonize bare rock or other barren surfaces. The fungal hyphae secrete acids that slowly dissolve minerals from rock, beginning the process of soil formation. And when the lichen dies, its organic matter contributes to the developing soil. Over centuries and millennia, lichens can transform barren rock surfaces into environments where mosses and then vascular plants can establish, playing a foundational role in primary succession. In established ecosystems, lichens contribute to nutrient cycling, particularly nitrogen cycling when cyanolichens are involved. The cyanobacteria fix atmospheric nitrogen, and when the lichen dies and decomposes, this nitrogen becomes available to other organisms. In boreal and Arctic regions, lichens can be the dominant ground cover, and they provide crucial food resources for animals. Caribou and reindeer depend heavily on lichens, particularly species in the genus Cladonia, for winter forage. In some regions, lichens constitute 80 to 90% of caribou diet during winter, when other food sources are buried under snow or unavailable. An acre of undisturbed land in northern Canada can contain 250 kilograms or more of lichen biomass after a century or more of growth, representing a substantial food reserve for herbivores adapted to digest these unusual organisms. Lichens are also extraordinarily sensitive to air pollution, particularly to sulfur dioxide and nitrogen oxides, pollutants produced by burning fossil fuels. The reasons for this sensitivity are not entirely clear, but likely relate to the lichen's physiology. They have no protective waxy cuticle like vascular plants do, absorb water and nutrients directly from precipitation and air, and cannot shed contaminated tissues. Pollutants accumulate in lichen tissues, disrupting photosynthesis and damaging both partners. In heavily polluted areas, lichens disappear, and lichen diversity declines with increasing pollution levels. This sensitivity has made lichens valuable indicators of air quality, biological monitors that integrate pollution exposure over time. Lichenologists can survey lichen communities and estimate air quality based on which species are present and absent, which are thriving and which are declining. The return of lichens to areas where air quality has improved, as sulfur dioxide emissions have declined, following clean air regulations in many developed countries, provides visible evidence of environmental recovery, testament to the effectiveness of pollution control measures. Shift now, allow your mind to move from these composite organisms, these symbioses so complete that the partners form a new integrated whole, to partnerships that remain more loosely organized, where the partners maintain clear separate identities, where the relationship operates not through biochemical integration, but through behavior, through interactions that can be observed, that play out visibly on the African savannas. Picture a buffalo, a Cape buffalo, massive and dark, standing perhaps 5 feet at the shoulder, weighing close to a ton, horns curving down and then up. A formidable animal few predators will challenge. The buffalo stands grazing, pulling grass, chewing methodically, and on its back, perched along its spine, moving over its shoulders and flanks, are birds. Small birds, the size of starlings, with red bills or yellow bills, depending on species, moving nimbly across the buffalo's body, pecking at its hide, reaching down to probe between hairs, walking along the buffalo's face, even perching on its horns or disappearing momentarily into an ear. These are oxpeckers, and they are engaged in one of the most visible symbioses on Earth. A relationship that seems straightforward, seems like textbook mutualism, yet turns out upon closer examination to be far more complex, far more ambiguous. A relationship that blurs the line between cooperation and exploitation, between partner and parasite. There are only 2 species of oxpeckers in the world, both found exclusively in sub-Saharan Africa. The red-billed oxpecker and the yellow-billed oxpecker are members of the starling family, but they have evolved into a unique ecological niche, becoming the only birds in Africa, possibly the only birds anywhere, that live obligately in association with large mammals. For oxpeckers, the mammal is not just a feeding site, but an entire habitat. The environment where they spend most of their time, where they find food and safety, where they court mates, and sometimes even nest on the backs of their hosts. Different oxpecker species show preferences for different hosts, though there is overlap. Yellow-billed oxpeckers are found primarily on buffalo, zebra, and greater kudu, with buffalo accounting for over half of all sightings in areas where both oxpecker species occur. Red-billed oxpeckers associate with a wider range of host species, showing particular affinity for impala, but also found on giraffe, rhinoceros, warthogs, and various antelope species. The 2 species appear to reduce competition through this host partitioning, each specializing on different sets of mammals, even where their ranges overlap. The traditional explanation for the oxpecker mammal relationship is straightforward mutualism. The oxpeckers feed on ticks and other ectoparasites found on the mammals, removing blood, feeding ticks that can transmit diseases, that can cause anemia in heavy infestations, that weaken their hosts. The mammals benefit from this cleaning service, from having parasites removed, while the birds gain a reliable food source. This seems simple, beneficial to both parties. A clear example of mutualism, where each partner gains something of value. And for a long time, this explanation was accepted without serious question. Oxpeckers were called tick birds, and their role as cleaners of large mammals was presented in textbooks as a classic example of interspecific cooperation. Farmers and wildlife managers valued oxpeckers, believing they provided pest control services, reducing tick burdens on cattle and wildlife. But in 1999, a researcher named Paul Weeks conducted a careful field experiment in Zimbabwe that challenged this simple story. He excluded oxpeckers from some cattle while allowing them access to others, then compared tick loads between the 2 groups. The results were surprising. Cattle with oxpeckers did not have fewer ticks than cattle without oxpeckers. In fact, tick numbers were similar in both groups. Closer observation revealed why. Oxpeckers preferentially fed on ticks that were already engorged with blood, ticks that had fed on the mammal and were full, rather than removing young ticks that had not yet fed. The blood inside the tick appeared to be the valued resource. An engorged tick provides far more nutrition than an unfed tick. and oxpeckers seem to be optimizing their feeding efficiency by selecting the most rewarding prey. This means they were removing ticks that had already done their damage, had already consumed blood, rather than preventing tick feeding. From a parasite control perspective, this is far less beneficial to the host than removing ticks before they feed. Moreover, oxpeckers do not feed exclusively on ticks. They also consume blood directly from their hosts. They reopen wounds, picking at scabs, and preventing healing, feeding on the fresh blood and serum that seeps from these maintained wounds. They consume mucus from nostrils and eyes, saliva from around the mouth, earwax from ear canals. They eat dead skin and damaged tissue. Some of this feeding might be beneficial, cleaning wounds, perhaps helping to prevent infection, removing dead tissue that might otherwise slow healing. But the behavior of keeping wounds open, of deliberately preventing scabs from forming, suggests a more parasitic relationship. The bird is maintaining a resource, ensuring continued access to blood and serum, rather than allowing the wound to heal. In captivity studies, it has been demonstrated that red-billed oxpeckers can inflict visible damage on cattle hides, creating raw patches through persistent pecking and feeding. Whether wild oxpeckers routinely cause such damage remains debated, but the potential clearly exists. So the oxpecker-mammal relationship is not purely mutualistic. It contains elements of mutualism, particularly tick removal, but also elements of parasitism, particularly wound maintenance and blood feeding. The relationship appears to be context-dependent, varying based on tick abundance and possibly other factors. When ticks are abundant, oxpeckers focus on tick feeding, and the relationship is more mutualistic, the mammals receiving genuine benefit from parasite removal. When ticks are scarce, oxpeckers may shift toward more direct exploitation of host tissues, feeding on blood and wounds, and the relationship becomes more parasitic. Research by Tiffany Planton during her doctoral studies examined this variability, looking at how tick abundance influenced oxpecker behavior and the nature of the symbiosis. She found that when preferred tick species were abundant, the relationship functioned as nutritional mutualism, with hosts providing ticks in exchange for cleaning services. But when tick abundance was low, oxpeckers were more likely to feed on host tissues directly, shifting the relationship toward parasitism. This context dependence is fascinating because it suggests that symbioses are not fixed, not always one thing or another, but can shift along a continuum depending on circumstances. The same two species can have a relationship that is beneficial, neutral, or harmful depending on environmental conditions. Resource availability, the specific individuals involved. This flexibility challenges simplified classifications of symbioses into neat categories of mutualism, commensalism, or parasitism. Real biological relationships are messier, more dynamic, more responsive to context than abstract categories suggest. The oxpeckers and their hosts demonstrate this complexity, showing how a relationship can simultaneously contain cooperative and exploitative elements, how benefits and costs can shift with changing conditions. Despite these complications, phylogenetic analyses using data on oxpecker preferences for different ungulate species suggest that overall, the relationship is probably more mutualistic than parasitic. Oxpeckers show strong preferences for host species that harbor high tick abundance. They are found most often on the mammals with the most ticks. suggesting that ticks, not host tissues, are the primary food source driving host selection. If oxpeckers were primarily parasitic, feeding mainly on blood and wounds, we might expect them to prefer hosts with thinner hides. Hosts easier to pierce and injure, but hide thickness does not predict oxpecker preferences. The birds do not disproportionately select thin-skinned hosts. This suggests that while oxpeckers do feed opportunistically on host tissues, this is not the main driver of the relationship. The primary resource is ticks, and the primary benefit to mammals is probably tick removal, even if that removal is selective and not as comprehensive as might be ideal. Oxpeckers provide another benefit that may be equally or more important than tick removal. They serve as sentinels, as early warning systems for their hosts. Oxpeckers have excellent vision, far better than many of their host mammals, particularly better than rhinoceros, which have notoriously poor eyesight. When a potential threat approaches, whether a predator or a human, oxpeckers give alarm calls and fly up from their hosts, creating visible and audible signals that alert the mammals to danger. In Kiswahili, oxpeckers are called askari wa kifaru, guardians of the rhinoceros, a name that recognizes this sentinel function. For rhinoceros, which rely heavily on hearing and smell because their vision is poor, the presence of oxpeckers provides an additional sensory channel, eyes on their backs watching for threats. Studies have documented that mammals with oxpeckers react to danger sooner than mammals without oxpeckers, that the birds' alarm calls give their hosts advance warning, time to flee or prepare to fight. This benefit may be more valuable than tick removal, particularly for species like rhinoceros that face intense hunting pressure from humans. A few extra seconds of warning can mean the difference between escape and death for a rhinoceros confronting poachers. The relationship between oxpeckers and mammals also provides insight into the evolution of symbiosis, into how such relationships can arise and become more specialized over time. Oxpeckers likely evolved from ancestors that fed on insects, perhaps species that caught insects flushed by large mammals, or that fed on insects in grasslands where mammals grazed. The transition to feeding directly on mammals, to exploiting ectoparasites on the mammals' bodies, represents a significant ecological shift. It requires behavioral adaptations, the ability to land on and cling to a moving animal, to tolerate the animal's movements and reactions, to identify profitable feeding locations on the body. It requires morphological adaptations. Oxpeckers have sharp claws and relatively short legs that help them grip hide, and bill structures adapted for grasping ticks and probing between hairs and into crevices. These are specialized traits, adaptations for their unique lifestyle, and the relationship is obligate, at least from the bird's perspective. Oxpeckers are almost entirely dependent on their mammalian hosts. They feed almost exclusively on what they find on mammals' bodies, and they struggle to survive in areas where large wild mammals are absent. The use of pesticides to control ticks on cattle has led to dramatic declines in oxpecker populations outside protected areas, because cattle treated with pesticides carry few ticks, providing inadequate food for the birds. Yellow-billed oxpeckers in particular have suffered population declines and range contractions, becoming largely restricted to conservation areas where wildlife populations remain high. This vulnerability demonstrates the degree of dependence, the the extent to which the birds have committed to this ecological strategy. From the mammal's perspective, the relationship is facultative. They can survive perfectly well without oxpeckers. Before oxpeckers evolved, African ungulates dealt with ticks and other parasites through other means, through grooming, through wallowing in mud or dust, through allowing other bird species to feed opportunistically on insects around them. The arrival of oxpeckers may have provided some additional benefit, but it was not essential. The mammals tolerate oxpeckers, generally allowing them to feed and move across their bodies, but they are not dependent on them. This asymmetry in dependence is common in symbioses. One partner obligately dependent, while the other benefits but could manage without the relationship. Let your mind rest now. Settle into the understanding that symbioses come in many forms, occupy many points along continuums of integration and benefit, of cooperation and exploitation, of dependence and independence. We have explored partnerships where organisms merge into single composite structures, like lichens, where the distinction between partners becomes blurred, where a new emergent whole arises from the combination. We have seen partnerships mediated by complex anatomical structures, like nodules, specially constructed organs that house bacterial partners and facilitate exchange. We have examined partnerships operating through biochemical networking, like mycorrhizal fungi connecting plants through soil. And we have considered partnerships that are primarily behavioral, like oxpeckers and mammals, where the interaction is visible, observable, conducted through actions rather than through permanent structural or biochemical integration. These different forms of symbiosis reflect different evolutionary pathways, different solutions to the fundamental challenge of survival in a world where resources are limited, where cooperation can confer advantages that solitary existence cannot match. What unites all these partnerships, despite their diversity, is that they represent outcomes of natural selection operating on populations, shaping traits that increase survival and reproduction. Symbioses arise because individuals that form beneficial partnerships leave more offspring than those that do not. The mycorrhizal plant produces more seeds than the non-mycorrhizal plant because it accesses more nutrients. The legume with effective rhizobia fixes more nitrogen, grows larger, produces more seeds. The coral with abundant zooxanthellae grows faster, contributes more to reef structure, achieves greater reproductive output. The lichen persists in environments where neither fungus nor alga could survive alone, occupies niches unavailable to solitary organisms. Even the oxpecker benefits from its mammalian hosts, gaining access to food resources and safer foraging conditions than it might achieve alone, even as the benefit to the mammals remains less certain, more variable. In each case, natural selection has refined the partnership, has shaped the behaviors and structures and physiologies that make the partnership function, has created systems of exchange and coordination that span the boundaries between species, that create biological organization at levels above individual organism. The timing and coordination of symbiotic relationships also reveal remarkable sophistication. Consider the seasonal timing of oxpecker breeding in relation to the abundance of their food sources. Research in Kruger National Park has shown that red-billed oxpeckers time their breeding to coincide with seasonal peaks in tick and fly abundance. The pre-breeding molt requires protein, and this molt occurs when scaled ticks become abundant following the rains. Later, when horse flies emerge in greater numbers, The protein-rich flies provide resources that allow oxpeckers to initiate breeding, to produce eggs and rear young. This synchronization between oxpecker life history and the seasonal dynamics of ectoparasite populations demonstrates how deeply embedded these birds have become in the ecology of their hosts, how their annual cycle is tuned to the rhythms of mammalian parasite communities, how what might have begun as opportunistic feeding has evolved into an obligate lifestyle requiring precise temporal coordination. The efficiency of oxpecker feeding is also noteworthy. In captivity experiments conducted in the early 1980s, researchers demonstrated that red-billed oxpeckers could reduce adult tick populations on cattle by up to 98%, a dramatic reduction showing the birds' potential as biocontrol agents when they focus on tick removal. Analysis of nestling diet revealed that ticks accounted for nearly half the food fed to young birds, with hair and host tissues comprising about 35%. This dietary composition varies between oxpecker species. Yellow-billed oxpeckers, with their larger bills, are better able to handle the larger adult ticks, while red-billed oxpeckers with smaller bills are more efficient at capturing smaller ticks and appear to supplement their diet more with non-tick items. These differences in feeding capabilities may contribute to the host partitioning observed between species, with each oxpecker species exploiting slightly different resources, reducing direct competition even when they occupy the same geographic areas. The mobility of oxpeckers between hosts is another fascinating aspect of their behavior. DNA metabarcoding studies analyzing oxpecker fecal samples have revealed that individual birds move between multiple host animals within relatively short time periods. 32% of samples showed genetic evidence of at least 2 different mammal host species, indicating that oxpeckers do not remain exclusively with a single individual mammal, but move opportunistically between hosts, presumably tracking resource availability. A bird might feed on an impala in the morning, then move to a buffalo in the afternoon, then to a giraffe the next day. This mobility allows oxpeckers to exploit patchy resources efficiently. If one host has few ticks or becomes intolerant of the bird's presence, the oxpecker can simply move to another host, searching for better feeding opportunities. This flexibility is essential for a lifestyle based on resources that vary unpredictably across individual hosts and through time. The broader ecological implications of symbiosis extend far beyond the immediate participants. Mycorrhizal networks support forest resilience and biodiversity, influence which plant species can coexist, affect carbon cycling through ecosystems, nitrogen, fixing legumes in rich agricultural soils, support crop rotations that have sustained farming for millennia, reduce dependence on energy-intensive synthetic fertilizers, whose manufacture and application contribute significantly to greenhouse gas emissions and water pollution. Coral reefs built through the coral-zooxanthellae partnership support thousands of species, protect coastlines, provide protein for hundreds of millions of people. Lichens contribute to soil formation, provide winter forage for caribou herds that support indigenous communities, serve as biological monitors of air quality. Oxpeckers, despite questions about whether they are mutualists or part-time parasites, may help control tick populations and disease transmission in wildlife, and their sentinel behavior provides genuine protection for endangered species like rhinoceros. The partnerships we have explored are not isolated curiosities, not merely interesting biological phenomena. but fundamental components of how ecosystems function, how nutrients cycle, how energy flows through biological communities, how diversity is generated and maintained. Understanding symbiosis changes how we see the natural world. It moves us away from a view of nature as primarily competitive, as a war of all against all, toward recognition that cooperation and mutual dependence are equally fundamental, equally important in shaping life's diversity and complexity. It reveals that boundaries between organisms are porous, that what we identify as an individual may be a community, that what appears to be a single species may be a partnership. It shows that evolution operates not only through competition, but through cooperation, not only through selfish exploitation, but through exchange and reciprocity. This understanding has practical implications. In agriculture, it suggests working with symbioses rather than disrupting them, maintaining mycorrhizal networks and nitrogen, fixing partnerships rather than depending entirely on external inputs. In conservation, it highlights the importance of maintaining entire communities of interacting species rather than focusing narrowly on individual charismatic species, recognizing that saving the coral requires protecting the zooxanthellae, that supporting caribou populations requires preserving lichen communities, that oxpecker conservation depends on maintaining healthy populations of wild ungulates with their associated tick communities. The partnerships we have explored also raise profound questions about the nature of individuality, about what it means to be an organism. If a lichen is not a single species, but a community, if a coral cannot survive without its algae, if a legume depends on bacteria to provide essential nutrients, if a tree is connected through fungal networks to dozens of other trees, then where do we draw the boundaries of the individual? What is the appropriate unit of biological organization to study, to understand, to protect? These questions have no simple answers, but they push us toward more holistic, more systems, level thinking, toward recognizing that organisms are embedded in webs of relationships, that their survival and reproduction depend not just on their own traits, but on their interactions with partners. Competitors with the physical environment, with the entire community of which they are part. This ecological perspective, this recognition of interconnection and interdependence, may be essential for addressing the environmental challenges humanity faces, for understanding how ecosystems respond to change, for predicting the consequences of our actions, for finding living within the biosphere rather than simply exploiting it. As you rest now, as you allow these stories to settle into your understanding, remember that these partnerships are all around you, operating continuously, mostly invisible, mostly beyond your awareness, but fundamental to the world you inhabit. The trees outside your window are connected underground through fungal networks, exchanging resources and signals. The air you breathe contains oxygen released by plants and algae, many of them partnered with bacteria or fungi. The food on your table includes legumes enriched by bacterial nitrogen fixation, crops that may depend on mycorrhizal partnerships. The leather and wool and other animal products come from herbivores that host complex microbiomes, communities of symbiotic organisms in their digestive systems that allow them to digest plant material. You yourself are a symbiosis, your body hosting trillions of bacterial cells in your gut and on your skin. microorganisms that help you digest food, that protect you from pathogens, that produce vitamins you cannot synthesize yourself. Symbiosis is not rare or exceptional. It is everywhere, operating at every scale. From the molecular partnerships between cells and their mitochondria and chloroplasts, to the community-level networks we have explored tonight. It is a fundamental principle of biological organization, as important as competition or predation, or any other ecological interaction. And understanding it, appreciating it, may be key to understanding life itself, to seeing the world not as a collection of isolated individuals, but as an interwoven community, a biological network where cooperation and connection are as fundamental as conflict and competition. If you enjoyed this exploration and would like to support the creation of more content like this, please consider liking this video and subscribing to the channel. Your support helps make these journeys through science and nature possible. Rest well, and may your dreams be filled with the quiet marvels of nature's partnerships.